we should recognize that when we talk about, when we're referring to mitosis, we're talking about the duplication of the genome of that organism, and then like the splitting, if it has a nucleus, the division of that nucleus. Right? So we're talking about replicating its genome, replicating its instructions for a new generation of that species, or for the next generation of it, from parent cell to daughter cells, if you want to think of it like that. Meiosis is going to be more along the lines of the creation of haploid cells. There's diploid and haploid cells. The diploid cells are cells that have the complete set of chromosomes. And for humans, that'd be 46. A haploid cell, that would have half that amount. And those are also known as the sex cells, so like your sperm and egg cells. We'll see that happen again in multi-celled organisms that go through sexual reproduction, right? So we'll look at meiosis and karyotyping some things along the way. But first, mitosis and some cell regulation ideas. All right, so in the first segment of Unit 6, we're going to look at kind of like the growth and division portion of the cell cycle. And a couple of things for us to kind of think about as we move through this is, why are cells so small? I mean, if they're just going through the process of really kind of growth and then maybe creating some offspring, like why do cells at their level, like why divide? I mean, why not just get bigger and bigger, right? So we'll look at that a little bit today. And also consider the fact that you yourself, being composed of like trillions and trillions of individual cells now, originally were just the union of two haploid gametes to form a single diploid cell, right? That eventually divided, multiplied to being what you are now. And what makes you up, the instructions that code for you, are absolutely unique to you. Right? There is no one else before or after that will have the same genetic code that you have. Right? Even twins. Okay, so it's rather interesting. It's rather interesting, and it truly means that you are a unique and special individual, right? So we're going to think about that as we move through this idea of mitosis. All right. Cell division, we can kind of look at it in a couple different ways. First, on the side of multi-celled organisms, these will be organisms that have specialized cells, right? Some cells that take care of, their cardio cells and bone cells and nerve cells and stuff like that. So we have specialized cells to take care of different tasks for us. They have two different types of cell division. One, mitosis, right? This is to make more what we would say our body cells. More technically, they're called somatic cells. And they're simply like the things that you kind of make more of to maintain yourself. Right? Like in the event, if you cut yourself, making more skin cells to heal that cut. In order to reproduce, then, we need to, we could provide part of our genome to then, meet up with the haploid cell of another special individual, right? So meiosis would be the division process that we would go through to make those sex cells. Right? And because I truly think that, ladies first, the example here would be the egg cells. If you're a single-celled organism, right, all of your functions have to happen in that one cell. So we usually see, not in every case, but many cases in a single-cell organism, they will depend on mitosis for asexual reproduction. Right? The example being binary fission. In binary fission, simply we see a duplication, a replication of the genome that makes up the organism, along with some of the organelles as well that they may contain, if it's eukaryotic. And it'll simply make almost like an identical copy of itself. Right? So from one parent cell would emerge two daughter cells. They would refer to that. So an example of an organism that does that would be a simple amoeba. So I threw this little image on there. And so this amoeba is a single-celled eukaryotic organism. It's going to have a cell nucleus. It's going to have all the things associated with a eukaryotic organism, but it's going to go through binary fission. It does not exchange genetic information with another amoeba to make more amoeba. Prokaryotic cells first. Their cell cycle is a little more simplistic. First, they're going to go through growth, which is pretty obvious. And then they're going to go through mitotic cell division, or mitosis. And we usually term this binary fission because they're going to make doubles of things and then split in half, split in two. So the steps of this is one, a single chromosome, right, with all its genes are replicated. So in a prokaryotic cell, it does not have what? What are we lacking in that prokaryotic cell? Yeah, they don't have their DNA contained in a nuclear membrane like eukaryotic cells would. So they just have this kind of jumbled rubber band of genetic material, like a single chromosome in a loop, usually, all kind of squished in. And it's going to make a copy of that. It's going to duplicate that or replicate that chromosome or that genetic information. They're going to move those things to the far end of this growing, elongating cell. And then once that has been achieved, they're going to pinch off in the middle. Right? And now you have two daughter cells, identical genomes. You have a single cell prokaryotic organism here with its double membrane. It has a looped chromosome. It makes a duplicate of that. Goes to far ends of this elongated cell and then pinches off in between. We have a growing, the cell membrane closes in around in the middle, and we have two daughter cells. So we might ask ourselves, well, wait a minute, like why do we have to divide it all anyway? Why not just continue to grow larger and larger and larger? Why not? That's a good question. Right, so there's a couple things that we have to consider when it comes to limiting cell growth or really answering the question why a cell divides. Right, there are two major reasons here. The first, DNA overload. Right, so what do we mean by that? Right, the 3 bullet points kind of tie into this. One, the DNA is really like the control center of the cell because it's going to dictate like how things are made and how things are kind of done. Right, so that's one part of it. The second part then is as the cell gets larger, it's going to need that information accessed more and more often. Because if you just continue to grow larger, then obviously you're going to need to have more of the enzymes made and the proteins made and more cell membrane made and more Golgi apparatus and more of the other organelles that are already a part of your functioning cell. Well, that information is locked into the DNA, so we got to access it and use it to code for the replacement or building more things. And the problem will be that DNA just simply can't meet the demands of the cell. There's only you have a good length of information, but the gene that codes for a specific protein is located on a certain chromosome, and if it's already being, if the information is already being decoded from that, you can't decode it at the same time. You have to wait for that to be done. So if the cell keeps getting bigger and bigger, we have a problem. Right, so that's one reason why we just don't see like ginormous cells, like cells the size of elephants or whatever, which would be kind of cool, but yeah, that would be kind of neat. What is that huge thing? It's an amoeba. Oh my gosh, it's crazy. It's as big as a bus. In fact, there's a bus inside of it and engulfed it. Look at the kid screaming. Hey, is that my sister in there? Right. The second reason is the exchange of materials. We have to try to exchange materials through the membrane, and then we have to try to deliver them into a larger volume. We continue to get bigger and bigger and bigger. And so, in your notes, the last thing to mention here is that as the surface area increases, that ratio of surface area to volume decreases. And so it's going to take you a lot longer to try to get things delivered to the cell because the cell's not just 2 dimensions. You can pour back, left and right, but also up and down too, right? So you have 3 dimensions of space contained inside that cell to work and deliver things in. So growing bigger is not going to be a very efficient solution, right? What if we consider a cell being a single unit cube? right? So this is like a unit square, right? So it has size, height, width, and depth of 1 unit, and that means that it has a surface area of 6 square units, 6 sides to a cube, and it also would then have a total volume of 1 cubed, right? If we evaluate the surface area versus the volume that area encloses, it would be a 6-to-1 ratio. That's pretty good. That means that we have a lot of surface area exposed and a closing around a very small volume. So if we need to exchange things through the cell membrane and then transport them to some place in the cell, we can do that pretty well and pretty quickly. Right? That's like our single classroom cell. But what if we decide, that's mitosis stuff, forget it. We're going to get bigger and bigger. So we grow to a size of 5 units, height, width, and length. That means that we have now a surface area of 5 times 5 times 6 sides, right? So that would be 150 square units, right? And then our volume would be 5 cubed, or 125 cubic units. If we evaluate that new large cell, like the one that includes the courtyard, for the volume that is enclosed by that surface area, we find that we drop now to a 1.2-to-1 ratio, which means that things can we have surface areas there for things to move through, but as far as like the volume to transport things in, it's a lot more volume to handle per amount of surface area. So things will take a little bit longer. If we then say, all right, well, this being a large cell is not a good idea, we'll go through cell division. We can divide ourselves into still staying at 1 unit cubes, but we'd be 5 by 5, so 25 times 5 set of them. So we'd be 125 little cubes of that 1 by 1 by 1 dimension. That would mean that our surface area is now going to bloat to 750 square units because we have 6 sides to each of those 125 cubes. And our ratio is maintained. Right? Surface to volume ratio is 6-to-1 again. Happy day. Right? So yeah, I think the actual demonstration activity is a little bit more entertaining, for sure. Right? But for those people who are more mathematically inclined, you're pretty geeked out about this too. But the takeaway messages are, 1, the volume of a growing cell increases more quickly than the surface area would. And 2, if the cell gets too large, we can't import and export stuff as efficiently as we would if we were smaller. All right, so we got to divide. All right, so for prokaryotic organisms, division has 2 major purposes of growth and reproduction, right, through binary fission. Even single-cell organisms, I guess, could fall into that category. But for eukaryotic organisms that are multicellular, we need it for 2 different reasons. One being growth still, but the second being to repair as well. Right? So for growth and repair, we want to grow and make more of these somatic cells that make us up, because we have certain cells that do certain jobs, and we need to make more of them so we can keep doing those special jobs. We need to make a copy of this information that the cell contains. It's easy to do a web search on chromosomes, genes, human genome, whatever, and you're going to see images flash up on your screen like what we see on the left-hand side here. This classic X-looking chromosome, right? Or some people would say that, some people would call this a gene. That's not correct. So what we need to make sure we understand is that this is a chromosome that has been replicated already. It's densely packed and ready for cell division. Like ordinarily, when a cell is not preparing for cell division, you would not have this right-hand side of it. Right? It would just be this little narrowing part. If you were to condense it, it'd just be just the left-hand side. That's it. Right? It's still one chromosome. This is still one chromosome. It's just been duplicated. Right? A gene is a sequence of DNA that codes for a particular protein. This single chromosome has many, many genes in it. Okay, so just be aware of that. It's easy for you to get confused on the terminology and call that a gene. It's not. It's many of them. It's hundreds of genes, potentially. Could be like hair color and eye color and skin pigmentation code, genes. There's just a myriad of things could be all programmed into that one chromosome, right? So just make sure you understand that. So ordinarily, it's going to be in chromatin form. It's going to be all strung out like spaghetti, and the chromatin is in this strung out form, and we can't see it with a microscope yet. We can see these. We can see the condensed form of it, and it's going to go from a stringy, like chromatin-like form, where you have this DNA strand wrapped around a protein known as a histone, which then gets wrapped around a more larger structure known as a nucleosome, and then those guys are super coiled and packed to make it look like this chromosome. Right? So we'll move ahead a little bit and try to clean up a little bit of the mess. Now, along the way, we're going to be thinking about the question of how many chromosomes does a human have? You might know offhand. You may have looked ahead in the notes already. Just hang tight. We'll answer that question in a moment. All right. So a chromosome is really DNA and packing proteins that have been super packed and ready for mitosis, ready for cell division. Right? A chromosome has 2 telomeres. Right? So if this has been duplicated, but if we ignore the right-hand side, it has a top and bottom telomere. It has a narrowing in it known as a centromere. It's in the center of this chromosome, right? And we'll talk a little bit more as to like what this actually is put together. A single side of this is known as a chromatid, and so this is a copy of that chromatid. So they'd be known as sister chromatids. The 2 of them together, they are sister chromatids because they have the same genetic information. Right, but we'll get into that, I think, a little bit more as we move ahead slightly. In your note handout, there's several diagrams for you. One is like labeling the parts of a typical chromosome, right? So we could easily label the telomere. Sometimes people will refer to these as the arms of a chromosome, the centromere as well, and then a single side of this is a chromatid. Both of them together would be sister chromatids. Right, but I think we'll dig into that a little bit more again as well. So don't panic too much. So to go from a unreplicated to replicated DNA, right? In the nucleus, we'd have one of our chromosomes, known as a chromatid, would go through a process of replication. It's going to synthesize itself, right? So it makes a copy, right? The 2 of these are identical in genetic information for the most part, and they represent one chromosome, right? So it's a copy of that one chromosome. And this could be the chromosome, one of the chromosomes that you inherited from your parent, your paternal genetic donor, your father, or it could be from your maternal genetic donor, your mother, right? But either way, one of those chromosomes gets duplicated, and then it's going to pack up. Right? So this is going to be where we start getting ready to go through mitosis. And it'll form that typical X-looking condensed structure with the centromere in the middle, your telomeres are the branching arms, and these are sister chromatids now. Still one chromosome, 2 sister chromatids. After we go through mitosis, we will pop these guys apart, and they will go to the respective cells. And there will now be 2 chromosomes. They're the same chromosome, but they've been now split from, the sisters have been separated, and they are now in their own respective cells. Okay, so that's how we go from a like stringy like chromatid to a one that has been duplicated, has gone through DNA synthesis, to the condensed structure, to separate cells. Okay, so that's where we see the different images, I think, play out. Okay, so this is another look at the same idea. These sister chromatids will separate during cell division. So in a brand new cell that's not worried about going through mitosis and duplicating, making more of itself, or going through cell division, we would find that if we were to coil up that chromatid, it would just be one half of that X-like structure. When we are ready for cell division, we will make a copy of that, and they will be held together at the centromere, and these would be sister chromatids. And then when we actually go through mitosis, we'll pull each sister to either end of the cell just prior to dividing and then through cytokinesis. All right, so just another look at the same sort of idea. All right, now as far as like humans go, right, we have 23 pairs of chromosomes. Right, so if we have 23 pairs of chromosomes, that means we have 46 chromosomes altogether. 23, again, from your paternal donor, 23 from your maternal donor. Or do you prefer mom and dad? I think paternal donor and maternal donor sounds cool, sounds more sciencey, but whatever. All right, these homologous chromosomes, or homologs, will be our like coding for like hair color and eye color, and you need a couple eyeballs, and you probably need a heart someplace, and so like all this information that makes up what you are generally, right? And then the 23rd pair would be where sex-linked chromosomes would come into play, right? So the sex chromosomes are either X's and Y's. If your karyotype, like if we look at those images at the bottom, those are karyotypes. A karyotype is simply like a picture of, an image of the chromosomes that make up an individual, right? So if you have an X and a Y sex chromosome, you will be male, and if you have an X and an X chromosome, you will be labeled as female. The way that your body grows and develops, provided again based upon what your genome contains, would put you in the category of providing the sperm gamete. If you have X and Y, it'd put you in the category of providing the egg or the ovum. If you have X and X sex chromosomes. Right now, the reason I put these two images up though is because this could be the same person. Right? This could be the same person. What's different is on the picture on the left, this is someone's cells that has not replicated its DNA yet. Right? So this could be like a brand new cell. Hasn't replicated or synthesized its DNA yet. Has 23 pairs of chromosomes. Right? Total of 46, half from mom, half from dad. Right? And if we look at the picture on the right, again, we have 23 pairs. Just we now have each which was a single chromatid has gone through synthesis to make a copy of itself. Right? Locked together, stuck together at the centromere. Right? So I don't want you getting into getting confused and thinking that like each one of these is a chromosome. So now when I look at the picture on the right, under number 1, there are 4 chromosomes there now. No. There's still just 2 chromosomes. One from mom, one from dad. They just have made a duplicate. They're ready for cell division. Right? So it's possible when if you do some web searching or you look in your Flexbook on material, and they're talking about chromosomes and karyotypes and stuff, don't get confused about if you see images where they are not making the X's, they're not making those X shapes, right? That they don't have their sister chromatid yet. It's still just a gene from your mom, a gene from your dad, a pair of homologous chromosomes. Okay. Different species have different numbers of them. We have 46 different chromosomes. Right? 23 from mom, 23 from dad. And other organisms still have varying amounts. Like your typical housefly has 12. Frogs have 26. Dogs have 78. And the adder's tongue fern, which is this picture here, right, can have up to 720 chromosomes. There are organisms like this one where they'll go through mitosis, but not cytokinesis. They'll duplicate their nucleus, but not split into new cells. So that's why in this plant, it's possible to have a single cell that has 4 to 6 different nuclei in it, right? Which is pretty weird. But it's called polyploidy. Okay, so the cell cycle really is just like repeated interphase and then mitosis, and then interphase and mitosis, and then interphase and mitosis, like over and over and over. Right, so you should have a graphic similar to this in a section of your notes. You can feel free to add things to this, mark it up, and then add information as we move through, just like anything else. But we're going to look at the major phases of the cell cycle, and then we'll focus in on mitosis. Right, so mitosis is just one small wedge of the entire cell cycle for a eukaryotic cell. All right, so we should make mention of the different phases, and then we can sketch them into our diagram. Right, the G1 phase of a cell cycle is going to occur right after cytokinesis. Right, so this is just after the cell has been made, a new cell has emerged, and in your graphic in your notes, it'll be the top light gray shaded section. Right. That's where cell growth is going to happen. Right? Cell growth and maturity, we're going to be making proteins, we'll be duplicating organelles, mitochondria will be going and making copies of themselves, they're going through binary fission on their own. It's just the cell's doing what it's supposed to do. And a multi-celled organism, it could be, it could be skin cells, making some new skin cells, growing, maturing in the dermis, eventually going to die and make its way to the epidermis to protect us from invaders. But for right now, it's growing and carrying out its typical routines and functions. Now, when we move into S phase, right, that's going to be where the DNA will get replicated. That's where DNA synthesis happens. And this is where each chromosome will make its sister chromatid. A single chromosome will make a copy of itself. Well, it not on its own. There's a process to it. That's later on. But for now, we're just going to say that the chromosomes are duplicated. And that's going to be in the lighter wedge to the right-hand side of your pie diagram that you have. And then the G2 phase is another, they call it a gap. Like the G1 is first gap, G2 is second gap. But I don't know, I think most people that I've talked to recently, like my prof several years ago, and I was talking with Elser, I mean, you can also kind of call these growth phases. I mean, I don't know if gap is necessary. I think they call it gap because you're not doing anything with the DNA so much. Like you're not replicating it, you're not looking at making a duplicate of the nucleus at this point. That's kind of the focus that we're looking at to hone in on. But so we have another G2 phase, and it's going to go also through some checks. Like cell cycle's got to check some things out to make sure that the DNA was replicated okay. We have the numbers of things are duplicated all right, and everything's ready to go for mitosis. Right? It's when the conditions are right, when the chemistry in the cell is correct, when those indicators are present that says everything is good to go. It's like if you're watching like a SpaceX rocket launch, right? If you watch those live on YouTube, like there's a series of checks they go through. It's like, now at this point, the computer has assumed control of the launch vehicle, countdown at 10, 20, and counting. That's what we're getting to. Right? G2 is kind of going through all those steps, making sure everything's ready to go for the trip. All right, and finally, M phase. Now, I believe in your flex book it separates these two. The next couple things that happen. That mitosis, by definition, is the division of the nucleus. But after that occurs, it's followed up with cytokinesis. Older textbooks actually group these together. I think it's proper to keep them separate because mitosis is a series of complex steps that do result in the division of the nucleus. Whereas cytokinesis is where you actually divide the cytoplasm, like you actually then finish the job and divide into daughter cells. All right, so we can kind of fill this guy in, and we can mention that interphase includes G1, S, and G2. And then you have M phase, which is separate. Cells will spend most of their life cycle in interphase. Cells divide at different rates. So, I mean, if we're talking about cells spending different amounts of time in interphase before they go through mitosis, mitosis is really to create a brand new cell from what you have existing. Right? The skin, lining of the mouth, the stomach, very fast, almost daily. We're looking at, daily we're seeing new cells being made, actively going through mitosis to repair the cells that are lost. Like whenever you put that sock on or put your clothes on and take them off and rub things on your hands and rub the skin, you're pulling dead skin cells off the surface of your body all day long, and those need replaced. Right? So when you eat stuff, process things in your stomach, those environments, a lot of wear and tear on the biology, and needs to be fixed, needs to be repaired or replenished. So fast. Bone and muscle, not so much, about weekly. Liver, lung, hearts, and even kidney tissue, looking at on a yearly basis of cell mitosis occurring and looking at replacement of those cells and repair. Brain and nerve cells, never. There is a lot of research in trying to coax like brain and nerve cells out of this G0 phase to make them make more of themselves or repair others or replace those that have been damaged. That would be a very big breakthrough if they were able to somehow reactivate these somatic cells. All right, so what about this G0 phase then? Only certain cells do it. Red blood cells. Once the red blood cell has been made, the red blood cell will serve its purpose until it's untimely or demise, until it's no longer functional as a red blood cell, and then gets metabolized and broken back down. Nerve cells also do this. So they just exit the cycle. Some things that you read about this would say that they kind of just stay in G1 like forever. They don't move towards wanting to make more of their genetic information, and therefore they don't go through the movement into S phase and G2. They stay in G1. All right, so we talked about G phase, S phase, and G2 a little bit. We talked about cells that park themselves in G1 and maybe take a hiatus in G0 and kind of just hang out and do their job. But we're going to look at M phase, right, and all these steps that fall into mitosis and then leading into cytokinesis. All right, there are two parts. Mitosis is that division of the cell nucleus. You can remember that by the acronym PMAT, right? So prophase, metaphase, anaphase, and I've always called it telophase. I heard someone else calling it telophase. I don't know if there's much one way is more right than the other, but I think more people call it telophase. All right, so prophase, metaphase, anaphase, and telophase. And then also cytokinesis. We're going to state that as a separate phase or separate step in M phase because it's really the cinching up and separation of the cytoplasm. And the cell membranes will get pinched out together and will bloop into two different cells. So This is going to happen after telophase in most cells. There are some cells that, some organisms that have cells that do not go through cytokinesis. We had mentioned that the Adler plant is one of them. And I don't know if you really could say that this is officially part of mitosis. It's a separate step, right? It's not really handling the duplication of the genome and its movement to another cell. Right? It's really the process of breaking the cell into two afterwards. All right. All right, so on we go. Prophase. And all my wonderful drawings. It's great. Okay, so the chromatin is going to make a copy of itself in S phase. That's already been done. So in prophase, as we leave G2 and go into M phase, the cell says, you know what, we need to divide. We're just too big. We've lived a good life. It's time for us to pass things on to another generation of cells. So we need to coil up and pack up our DNA. Right, so they're going to coil these guys up into the form of chromosomes, right? Those densely packed sister chromatids, and they're going to form spindle fibers that emerge from the centrosomes. Centrosomes are also known as centrioles, or centrioles. They will be like things that spindle fibers kind of wisp out from, and they migrate to where they're at opposite ends of the cell from one another. They're kind of like the highway coordinator or something to that effect. They're going to kind of orchestrate, they're the puppeteer, pulling the strings on the chromosomes and the cell, really, at this whole process. Right, the nuclear membrane will disappear, so the centrioles emerge, the spindle fibers start to form, and the nuclear membrane disintegrates. And we're going to then have these fibers known as kinetic core fibers link in to the motor protein that we call the kinetic core, right? That's in the centromere of your chromosomes. So where that tightened, like kind of connected area that the sister chromatids are stuck together at, that contains a little motor guy known as a kinetochore. Kinetics or kinesis means movement, right? So it's a little kinetochore that's going to help it move, and they're going to lock on or be attached to these special fibers. Or as we go through prophase, right? We have the centrioles. A pair of centrioles is known as a centrosome, and sometimes known as mitotic spindles. But they will start to move to opposite sides of the cell. And as they do that, the spindle fibers start to form. Some are kinetic core fibers that will link right in to the centromere of the chromosome. In that centromere, there is a protein motor known as a kinetic core, and it's going to help later on in the division process. Right, so we have that, but we also have polar fibers or non-kinetic core fibers because they don't connect to the kinetic core, but they connect from centriole to centriole to form like a framework so that they can pull and push and tug and stuff on these chromosomes to get them into position. Right, so prophase is all about that, building the structure to help facilitate the division of the cell, but more importantly, the separation of those duplicated chromosomes. Right. Well, these kinetic core fibers are really just fancy microtubules. We mentioned that back when we looked at the cytoskeleton of a cell. They're like a hollow tube made of the protein tubulin, and they are just really structural cables, really, if you want to think of it as such. Right? So the structural cables that we can then hook up or link up to a protein motor and use them as well as being connected to the centriole to help position these chromosomes, help move them into position. All right, so I found this really awesome simulation of how these guys all work. So it has some narration to it, so I'll try my best to provide the audio to you but without feeding back the system, and just kind of enjoy it for what it is.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions.
So this is your centromere and then your sister chromatids.
The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
So that will bring us to metaphase. When we get these guys lined up near the middle of the cell, it's known as the metaphase plate. And there's not like a physical plate there, but it's just like the region that's like near the actual middle of the cell, right? And also consider that in these drawings, I'm trying to show probably like 4 chromosomes that are lined up here. But you don't need to, for humans, there would be 46 of these guys lined up at the midpoint of the cell in 3 dimensions, okay? So if you took like a spear and cut a plane through the middle of it, you'd have 46 chromosomes lined up along that plane in 3 dimensions, right? So don't get stuck on like the other, they have to be in a straight line. They're not in a straight line, they're all coplanar. Would be the better way to think about it. 46 of them. So they'll line up at metaphase, and once they're lined up, as you saw in the animation, there's going to be a signal set saying, hey, you know what, we're actually in the right spot, we're at metaphase now, we're ready to rip the sister chromatids apart, pull them out away from one another. All right? And to do so, the kinetochores will start walking or pulling the sister chromatids apart from one another, heading towards the opposite side of the cell. All right? And that's going to bring us to anaphase. Right, so anaphase is going to be where our spindle fibers, the kinetochore protein motor, is actually going to walk or pull one of the sister chromatids away from the other and make its way towards the centrioles at either end of the cell. At the same time, those fibers that are polar, that go from one centriole to the other, are going to lengthen to try to stretch and elongate the cell. So we're trying to make room for what happens after mitosis. We're trying to make room for cytokinesis at the end. So again, I try to pick figures that have 4 chromosomes going on with them, and we'd have 46 of these guys lined up and being pulled to either side. All right, as you kind of sketch that into your notes, also I included a figure of a close-up diagram of a chromosome, a chromatid with its kinetochore protein motor embedded, and it's simply going to really motor its way along this microtubule that is that kinetochore fiber. And as it does so, on the back side of this kinetochore motor, it's going to like break off the tubulin that makes up that filament to be used someplace else, or maybe even cycled back to be used for cytoskeleton purposes, or maybe saved for things later on when the cell would divide again in its new life cycle. Right, but the movement of the chromosome would be like almost eating away at that kinetochore fiber as it moves its way towards the centriole. Is that okay? So a little more added detail to what you already may understand for mitosis and the steps. All right, so once we make our way to either end of the cell, it's telophase. I've been saying it wrong for over 30 years. Right? My biology instructor in high school called it telophase. I was instructed in it being telophase at the college level, and last evening I heard someone refer to it as a telophase, and then so I looked it up. I looked up what the pronunciation was, and it's a hard E, long E sound, so it's telophase. Who knew? Well, we know now, right? I guess you also talk about telephone. Yeah. So there you go. We now call them cell phones now, right? So I mean, there's no telephone. Celophones. Right, but cell phone has two L's, so I guess I don't know. But telophase. If I slip up and call it telophase, I beg your forgiveness. If you say call it telophase, okay, whatever. But it's like tomato or tomato, and they both are great on sandwiches and burgers and pizza and stuff. So whatever. So at telophase, we're going to have those chromosomes at opposite sides of the cell, which is the whole goal from when anaphase kicked in. Spindle fibers will be broken down and disappear. We don't need their use anymore. We don't have the need for them anymore. The chromosomes will start to unravel back into chromatin. The DNA will no longer be all tightly packed up as chromosomes. It'll go back to its chromatin format. Because why would that be? Well, because we're no longer needing to transport it, and the cell's getting ready to, once it finishes M phase, to go into interphase, where it's going to be using that DNA to read it and make instructions for building more proteins and more organelles. And then two nuclear membranes will reform around this now uncondensing chromosome, sets of chromosomes. So we're really close to having two new daughter cells here, but we're still one cell with two nuclei. So this would be the end of mitosis. The nucleus has been duplicated. Mission accomplished. All right, so after telophase, in most cases, not all, but in most cases, we'll be looking at going through cytokinesis. Right, to help cells do that, we're going to make use of another filament known as another type of fiber known as a microfilament. So microtubules are simply like structural strands, like cables. Microfilaments are fibers that actually can contract and relax. Right, the same fibers that would make up skeletal muscle tissue and build those tissues in. It's the same concept, same idea. Right, so we have a bunch of this actin protein ready to help constrict the cell membrane and close it down and pinch it off. So to where we have two daughter cells with their own separate cell membranes containing all that they are. This is known as a cleavage furrow, where it starts to clamp down and shrink down around the cell membrane, or rein that membrane in, where you almost can see like a dividing line where that cell is going to eventually separate. All right. So in animal cells, we see the cleavage furrow. This is an image from a microscopy view of a cell finishing up through cytokinesis. And then on the right-hand side, this is a plant cell. You can easily tell by the cell wall and the boxy shape, and you can kind of see the cell plate has formed. We'll use the cellulose to build in a cell wall at that spot. That cell plate is made up of some vesicles that have blobbed off from the Golgi apparatus and found their way to the midpoint between the two cells, or between the two nuclei. So both can be referred to as cytokinesis. It's just in an animal cell, we use that idea of a microfilament going to form that cleavage furrow and bringing the membrane close to where it can pinch off and separate. With a plant cell, the Golgi apparatus kind of blobs some vesicles in place and forms a scaffolding for the cell wall to be constructed and divide the cells in two. Now, what if you have a cell that does not go through cytokinesis? Right, that's a possibility. We mentioned that happens in that Adler fern, that it has over 120 chromosomes. At some point, cells that make up that plant can have up to 720 chromosomes. And the mention was that it does not go through cytokinesis. It goes through mitosis, so it duplicates the nucleus, but it does not divide the cell. We also have cells in our body that do that as well, particularly those that are involved with our skeletal system. Right, so we have osteoclasts. Osteoclasts are cells, large cells, multinucleated cells that break skeletal bone down. Their purpose is to help refine and repair the existing bone tissue or bone that you have. So if you, when your skeletal structure has like microfractures or just like wear and tear on it, these guys are going to go to that site. They're going to like latch onto the bone itself, and they have almost like a pit in the middle of them that will work on chemically dissolving the bone, so that a nearby osteoblast can build new bone after they're done. Right, so the question might be like, well, why would if you're just going to be breaking bone down, right? Why do you need such a large cell, and why does it need to be multinucleated? Right. So here's a couple, I think, good reasons as to why, and there are reasons that tie into difficulties we found with if the cell grows too large. Right. First, if you have more than one nucleus, you do not have a problem with information exchange. You can easily continue to read the DNA and make proteins and enzymes. That's one thing. In order to break bone down, it takes a lot of effort because it's a very strong, rigid structure. We know that we use it to walk around all day long. So it's going to take a lot of effort to break it back down and repair it. So that's one reason why we have a multinucleated osteoclast. Why such a large cell? Well, because you have more than one nucleus in there, you need more room. And also, the bigger the cell gets, we noted that the surface area also increases. That's going to allow you to exchange ions through that membrane at an increased rate. The osteoclast will kind of be like a flattened, pancakey looking cell that's latched onto the bone for the dissolving of the bone, and then osteoblast is a single nucleus cell that will go for repair to build the bone back. Usually really near or nearby where the osteoclast is. So the cartoon's kind of cool because you have the multinucleated osteoclast here, and yeah, he feeds on bone. That's what he eats while he breaks it down. We also see it in fruit flies. So not just isolated to our own osteoclast, but we see certain fruit fly in certain embryonic stages of their development. They are still multinucleated at some points. And then also, it can be occurring when we find tumors, in us and other organisms. That something in the cell cycle is not right, and a possible pathological process from that is that the cell goes through mitosis but not cytokinesis as it should. So anyway, we made that mention, and I thought it was kind of interesting to at least add this into the set so that way you're aware of that. We have some cells in our body that do mitosis but don't cytokinese.